The protein we have just watched emerge from the ribosome has not yet reached its final destination. Instead, many newly synthesized proteins are produced directly on ribosomes attached to an extensive membrane network known as the Rough Endoplasmic Reticulum. Our journey now follows one of these proteins as it enters this remarkable cellular factory. The ribosome remains firmly attached to the membrane while continuing translation. Rather than releasing the growing protein into the cytoplasm, it feeds the amino acid chain directly through a channel in the membrane, allowing synthesis and transport to occur at the same time. As the chain continues to lengthen, it passes through the membrane into the interior of the rough endoplasmic reticulum. This compartment, called the lumen, provides a carefully controlled environment where newly made proteins begin the next stage of their development. Crossing the membrane marks an important transition. The protein is no longer simply a sequence of amino acids. It has entered a specialized workspace where it will be folded, inspected, and prepared before it can function elsewhere in the cell. Inside the lumen, the flexible amino acid chain drifts freely through the fluid-filled interior. Left on its own, this chain could easily fold incorrectly, so the cell immediately provides assistance from highly specialized molecules already waiting nearby. Large helper proteins known as molecular chaperones now surround the emerging polypeptide. They do not determine the protein's final shape, but they prevent unwanted interactions and guide the chain through the complex process of folding into its correct three-dimensional structure. Gradually, the protein settles into a stable arrangement of helices, sheets, and loops. Every fold is dictated by the amino acid sequence produced during translation, allowing structure to emerge naturally from the information encoded within the gene. Only after reaching its stable conformation can the protein perform its intended function. At this stage, quality control systems inside the rough endoplasmic reticulum verify that folding has been completed correctly before the protein is allowed to continue its journey. With folding complete, we move deeper through the interconnected membranes of the endoplasmic reticulum. Ahead, the ribosomes gradually disappear from the membrane surface, revealing another region that serves a very different purpose. The rough and smooth regions are directly connected, forming one continuous membrane system. While proteins remain behind in the rough our path now leads towards smooth tubular membranes specialized for entirely different cellular activities. The landscape changes noticeably as the membrane loses its attached ribosomes. We have now entered the smooth endoplasmic reticulum, an interconnected network devoted primarily to lipid production, calcium storage, and chemical detoxification rather than protein synthesis. Unlike the rough this region is dedicated to building the lipids required for cellular membranes. Here, enzymes continuously assemble phospholipids, cholesterol, and other lipid molecules that allow cells to grow, divide, and maintain their internal compartments. As we move between these tubular membranes, enzymes embedded within the smooth work continuously to construct new lipid molecules. These freshly synthesized lipids become essential components of membranes throughout the entire cell. New phospholipids are inserted directly into the membrane as they are produced. In this way, the smooth endoplasmic reticulum constantly renews and expands the membrane system, supplying material that can later be delivered to other organelles. The smooth also acts as one of the cell's major detoxification centers. Specialized enzymes chemically modify drugs, alcohol, and other foreign compounds, making them easier for the body to eliminate while protecting the cell from potentially harmful molecules. Our journey now returns to the proteins that successfully completed folding in the rough Their preparation is complete, and they are ready to leave the endoplasmic reticulum to continue along the cell's secretory pathway. A small region of the membrane begins to curve outward around the finished cargo. As the membrane bends further, it encloses the protein inside a transport vesicle, creating a protective compartment for the next stage of transport. A coat of specialized proteins assembles around the forming vesicle, shaping it into a stable transport carrier. This temporary scaffold helps the membrane curve and separate efficiently from the endoplasmic reticulum. The vesicle finally pinches free from the membrane and quickly associates with the cell's cytoskeletal transport network. Guided by molecular motors, it begins moving through the crowded cytoplasm toward its next destination. Ahead of us, a new organelle comes into view, the Golgi apparatus. Here, the proteins and lipids produced within the endoplasmic reticulum will be further modified, sorted, and packaged before being delivered to their final locations throughout the cell and beyond.